I am Homo sapiens. The development of life on Earth has been a long and complex process. For billions of years, single-celled organisms ruled the planet and established complex ecosystems, which have been discussed in previous sections. After billions of years of single-celled life, multicellular organisms began to evolve. The first multicellular organism, which we can call an animal, on Earth is the sponge. Studying sponges provides significant insight into the beginning of multicellular life on Earth. Sponges, classified under the phylum Porifera, are considered the oldest multicellular animals. Fossil evidence suggests that sponges originated about 600 million years ago during the Precambrian era. Some recent research now suggests this may have been 800 million years ago, when the Earth's supercontinent Rodinia began to break apart. In fact, the process of Rodinia's disintegration is linked to the development of multicellular organisms, a topic I will cover in another video. Despite their simple structure, sponges have several important features that highlight their multicellular nature. Unlike more complex animals, sponges do not have tissues or organs. Their cells are loosely arranged, giving them an extraordinary capacity for regeneration. If a sponge is broken apart, its cells can regroup to form a new organism. Sponges often display asymmetrical irregularity, lacking the specific body plan seen in more advanced animals, meaning they have no fixed structure. A key feature of sponges is their porous body through which water flows. Sponges use this feature to feed through a process known as filter feeding. Their bodies have numerous small pores called ostia, through which water enters. With the water, small bacteria, plankton, and other microscopic particles are also drawn in. Inside sponges, there are specialized cells called collar cells. These cells filter water through their small flagella and capture food particles. These particles are then digested by amoeboid cells, and the nutrients are distributed to other parts of the body. Thus, sponges obtain their food through the process of filter feeding without any complex digestive system. The skeletons inside many sponges are made of calcium carbonate, silicon dioxide, or spongin, a modified collagen protein. Their shapes are adapted to make water flow more effective. Despite their simplicity, sponges play important roles in marine ecosystems as filter feeders. They help maintain water cleanliness and quality by removing microscopic particles and bacteria. Additionally, sponges provide habitat and shelter for various marine organisms, including small fish, crustaceans, and other creatures. Some fish, such as cardinal fish, particularly the bongai cardinal fish, breed exclusively in the shade of sponges. These fish are known for their unique breeding behavior, where they lay eggs in the shade of marine sponges and corals. This helps protect their eggs and young. If sponges were to disappear from the Earth, many marine species would face a crisis due to the interruption in their breeding. Marine sponges are generally not dangerous, but some species can be toxic and cause skin irritation or allergic reactions. Most marine sponges are harmless to humans, though touching them can cause skin irritation or itching. Additionally, some marine sponges have antibacterial and antifungal properties, which can be useful in medical research. Studying sponges provides deep insights into the evolution of life on Earth from single-celled to multicellular organisms. Their simple structure and genetic makeup offer clues about the mutations that lead to multicellularity. For example, sponges have genes related to cellular adhesion and differentiation, which are fundamental to the development of multicellular organisms. Furthermore, sponges demonstrate that a high level of complexity is not necessary for multicellular life. The biological success and adaptability of sponges in diverse environments illustrate that even a modest multicellular organization can have many advantages. Now, To answer a simple and curious question that must have come to everyone's mind, if a sponge is classified as a multicellular organism or an animal, why doesn't it look like one to the average person? The answer lies in evolutionary biology and is quite simple. Evolution occurs through the struggle to meet basic survival needs and adapt to the surrounding ecosystem. The early sponges didn't need legs because organisms developed in water don't require legs like land animals do. Although some very small sponges move around with the power of water currents in search of new places, they stop moving once they grow larger and find a suitable spot. Additionally, when sponges evolved millions of years ago in the marine environment, there were no large multicellular organisms on Earth that they needed to chase, like fish or crocodiles. Their food consisted of very tiny organisms, including bacteria, plankton, and other single-celled prokaryotic and eukaryotic organisms that drifted with the water currents and naturally came into contact with the sponge's pores. All the sponge had to do was filter these particles for its food. Given the conditions when sponges evolved, they didn't need to walk or swim. Despite having readily available food, sponges didn't receive enough energy to develop a large and complex body system. Therefore, the first multicellular organism, the sponge, is exactly as it needed to be according to the circumstances of its time.